regards to what is white matter, what is gray matter, and then the names of what we call it depending on where we are in the central nervous system or the peripheral nervous system. All right, so when we're dealing with the central nervous system, remember these two structures, the brain and the spinal cord, that is the CNS, okay? And there's two types of tissue, white and gray, okay? So white is myelinated axons, and we call those tracts when we bundle them together, okay? Gray matter, all right, a little different here, all right? This is going to be the unmyelinated areas and unmyelinated axons. Okay, so we're unmyelinated axons, but also areas on the neuron where there's no myelin. Okay, so we know that to be the cell bodies and dendrites. All right, so when we're in the central nervous system, we refer to, all right, the gray matter. This is like the shell of the brain there. That's the superficial region. That's the cortex. So we call that the if we're in the cerebrum, we call it the cerebral cortex. All right. If we're in the cerebellum, it'd be the cerebellar cortex. Okay. So that's going to be that superficial layer, that outer layer. All right. Now a nucleus is going to be internal to the gray matter, but it's going to be within, in most cases, we'll see it kind of buried in, all right, white matter, all right? So these areas of gray matter basically are a clustering of cell bodies, okay? And so in the central nervous system, we refer to those clustering of cell bodies as a nucleus. So knowing what we kind of know a little bit from when we were talking about the nervous system, when you have a cell body, all right, that's where our greater potentials are going to be generated, which will eventually, hopefully, if we make threshold value, that will get us to our action potential, okay? But the cell bodies are where we're going to find other neurons synapsing onto these cell bodies, all right? So keep that in mind because we'll get more into detail about that. But that's the nucleus. It's just going to be a clustering of these cell bodies, all right, in our central nervous system there. So here we can see, nice picture here. Here's the cerebral cortex on the outside. All right, that's our gray matter. And then on the inside, you'll see, all right, this grouping of just little islands of gray matter all over the place. And we refer to those as the cerebral nuclei. And those are going to be primarily cell bodies. All right, and then you can see here in the white matter, all right, we've got our commissural fibers going from side to side. That's going to be our corpus callosum area there. And then this white area coming down in between the putamen and the thalamus there, that's going to be our internal capsule running through here. Okay, we'll talk more about this stuff when we get in there. Now when we jump into the cerebellum and the brainstem, all right, similarly, all right, here's the cerebellum. You got the cerebellar cortex on the outside. That's that gray matter out here and over here. All right, and then you got your white matter on the inside. Brainstem, same thing. All right, you'll have our white matter more internally, all right, and the gray matter on the outside with little clusters of gray matter here. Those are the nuclei there. And then as we get into the most inferior portion of the brainstem, the medulla oblongata and the spinal cord, here you can see again, all right, white matter mixed in with the gray matter. Now, interestingly enough, when we get into the spinal cord, we flip-flop it, meaning the white matter is going to be more to the outside, and the gray matter is going to be more towards the inside. Okay? Where when we're in the brain, all right, the gray matter is more outside, and the white matter is going to be more internal. Check it out. Here's the white matter for the spinal cord. Gray matter is inside. Go back up to the brain. All right. Gray matter is on the outside. White matter is on the inside. See? Well, part of it will be because of some of the actual physiological functions that are going to be occurring when we get those tracks. We'll learn those tracks and how. But it starts to transition. If you look here, when we get, well, I jumped too far. All right. You can see, all right, how the white matter starts to increase in proportion to the gray matter. Still a lot of gray matter on the periphery, on the outer portions, and more of the white is internal, and that's going to be, this is the midbrain here, And then when we get down into the medulla oblongata, you can start to see now more of the gray matter is more internal and the white matter is starting to kind of surround it on the outer portions here. And then we get into the spinal cord. It's more, we're starting to transition to more myelinated axons because now we've got to send the message out. Okay. All right. So I want to talk to you about the coverings to the brain, the cranial meninges. All right. And these coverings are made up of connective tissue. All right. And they cover the brain and they cover the spinal cord. So essentially they're going to cover, all right, your central nervous system here. And what they do is a couple things. They will help to protect, support, all right, and separate parts of the brain, some parts of the brain from itself, right, and then parts of the brain itself from the cranial cavity there. Okay, so and inside of these meninges and some of these layers, because there's 3 of them, we have blood vessels. Okay, but most importantly, we got this stuff right here. Cerebral spinal fluid. I like me some cerebral spinal fluid. Cerebral spinal fluid helps to keep the brain buoyant. Okay, without cerebral spinal fluid, all right, your brain could, I mean, I'm exaggerating when I say this, it could slip out of the foramen magnum in the base of your skull. All right, that couldn't really happen, but it helps to keep it floating up inside your skull. Helps to nourish tissue, protect it. All right, so we'll get into more of the details. So our cranial meninges, there's 3 layers. All right, the deepest layer is the pia mater. This should be 1 T, by the way. That's misspelled. It should be mater. It's M-A-T-E-R, mater. All right, so pia mater, that means soft mother. Okay, the middle layer is the arachnoid mater. Okay, spidery mother. All right, and then dura, this is my favorite one. All right, that means hard mother. Okay, that's the outermost layer. So dura is the outermost. Arachnoid is the middle, and then pia is going to be the internal or the deepest layer for the meningeal layers here, okay? Again, it's connective tissue. We're going to talk about the different types. So the innermost layer is the pia mater. All right, it's made up of areolar connective tissue. It's very thin, and this actually literally will stick directly onto the surface of the brain, onto the nervous tissue, onto the cerebral cortex. Right, and it goes into all the nooks and crannies. So the pia mater, it will go down into the sulci, uncover all the gyri there. Okay, and it's made up of that loose areolar connective tissue. Okay, the next layer is the arachnoid mater. All right, the arachnoid mater, all right, is going to be the middle layer, so it lies deep to the dura mater, the outermost layer. All right, but in here, all right, we have a space called the subarachnoid space. This is largely clinically significant because in the subarachnoid space, that's where you find CSF, cerebral spinal fluid. So if you need to go and draw some cerebral spinal fluid on somebody because maybe they might have, does anyone know why you would take cerebral spinal fluid? Meningitis. Meningitis. Okay, so if you want to test, I mean, there's a couple other different, reasons why you would take it, but if you're suspecting, you've got to insert a needle into this space and draw out some of the cerebral spinal fluid here, okay? In this space here, all right, the subarachnoid space, all right, we have these little small, we call them trabeculae, but they're little extensions. So here's the pia mater, all right, here's the arachnoid mater, and these little extensions that just go from the arachnoid mater to the pia mater, we call that the arachnoid trabeculae there. All right, and then the space is located here. And that's where the cerebral spinal fluid flows through. Okay? So the arachnoid mater, the connective tissue that it's made up of is going to be, okay, this is why we call it arachnoid mater, because it looks like webs. All right? Collagen, all right, we know collagen, very tough, strong protein fiber, but also elastic fibers. All right? It gives it a little bit of springiness and elasticity. Okay? All right, so we will see, all right, there's a space between the dura mater and the arachnoid mater that is called the subdural space. All right, it is what's known as a potential space, which means that space is not normally there unless something not good is happening. If you rupture a blood vessel, you can bleed into that space. I don't know if you've ever heard of a subdural hematoma, all right? But if you rupture a blood vessel from some sort of trauma, TBI, traumatic brain injury, all right, that space can start to fill up with blood, okay? And then that potential space is now an actual space, and you can start to have neurological issues going on there, okay? All right, then my favorite one, the outermost layer, all right, the tough mother, all right, this is the Dura Mater, all right? Now here's the thing, it is made up of 2 layers of dense, irregular connective tissue. Now those 2 layers are mainly fused together in a lot of spaces, but in some areas, they're not fused together, okay? So that we name both those layers. You have the meningeal layer and the periosteal layer. Okay, the periosteal layer, periosteal, sounds like periosteum, that's right. This layer is going to be just deep, all right, to the periosteum there. It forms the periosteum on the inside of the cranial bones there. So, all right, this will be attached to, all right, the inside of the skull. And then the next layer of the Dura Mater is the meningeal layer. This will be the deeper layer. This layer will be in close approximation to the arachnoid mater. Okay? So like I said, they're normally fused, but in some cases they separate. And when they separate, they themselves have their own space. And so we call these spaces dural venous sinuses. Okay? And venous referring to deoxygenated blood. All right, that basically all the deoxygenated blood gets drained into these spaces, and then these spaces will drain the blood from your brain into your jugular vein. Okay. So then you have a space outside of the epidural, outside of the dura, and that's the epidural space. Again, it is a potential space, which means it should not be there. But in that space, we have blood vessels. And in some cases, you can have an epidural hematoma. Again, normally from some sort of trauma, and you start to bleed into this space. And you no longer have a potential space, you have an actual space. That's the epidural space. Okay, so I'll show you a pretty cool picture right here. All right, we're going to take a sample of the skull here. Oops, come on. All right, so we're going to go from the innermost, or excuse me, not the innermost, the outermost layer in. Here's the skin, or your scalp. All right, on the outside of it, here's that periosteal layer. Now remember, all right, our cranial bones are flat bones. Right? They don't have a medullary cavity. So here you have the cortical bone here, then you have your medullary or your trabecular bone on the inside. I remember that's that sandwich I was telling you about. Here are the 2 slices of bread, and here's the peanut butter and jelly on the inside. All right, that's going to be inside the flat bone here. Now, here on the internal surface, all right, the periosteal layer, all right, makes up the periosteum on the inside of your skull. Now, again, you can't see it, but between the bone here and the periosteal layer, that is where your epidural space would be. But again, it shouldn't be there as long as everything's okay. Okay, so you will have blood vessels in that space. If they rupture, then you can start to bleed into that space, and now you have that epidural space. It starts to take on shape because of the blood there. So the layer, all right, here's the first layer, the periosteal layer. Just deep to that, there's the meningeal layer. Okay, like I said, normally it's fused, but in some areas they separate, and they create this space here. That's a dural venous sinus, and that's where blood drains into from the brain into these spaces, and these spaces drain from your skull. Okay. All right. So here's the Dura Mater. Okay. Just below that, all right, you can see we have the subdural space, another potential space. Okay. And if it ruptures, or if a blood vessel ruptures and you bleed in that space, then it's no longer potential. Here's the arachnoid mater, all right, that middle layer. And then just below that, or deep to that, here is your subarachnoid space, not a potential space. All right, this is where the cerebral spinal fluid flows through. And then this pink line here is represented by the pia mater. You can see here's all that arachnoid trabeculae all through here. You'll have blood vessels that kind of just sit inside of the arachnoid trabeculae there. And then right below the pia mater, here's your gray matter. Here's your white matter. Questions? Not too bad. Okay. So let's talk about, since we're talking about the cranial meninges, I want to talk to you about the cranial dural septa. All right. Cranial dural septa are going to be where those two fused layers of the Dura Mater, all right, are found to divide or create partitions between certain regions of the brain, okay, or parts of the brain. All right, they're going to provide support. So these here you will have to know for identification purposes. The first one is the falx cerebri. This divides or is found in between your right and left cerebral hemispheres. All right, it's the largest of the two. Now inside of the falx cerebri, we find these two sinuses. All right, the superior sagittal sinus and the inferior sagittal sinus. Those are called dural venous sinuses, and that's where these two layers, all right, from the septa, they separate, and blood will flow into those spaces there. So I'll come back to the slide real quick, show you. All right, here it's easier to see on this one. Here's the inferior sagittal sinus. So here's the falx cerebri. Okay, it's made of that dense, irregular connective tissue, two layers fused together, and they literally separate, all right, the left hemisphere from the right cerebral hemisphere. Okay, so at the bottom portion of this, they separate a little bit and they create this little space. It almost looks like a vein, but it's not. Okay, it's not a vein because veins have valves. These sinuses do not have valves. All right. Then at the top here, you can't see it very well, but at the top here, from front to back, all right, there's another space here, and that's the superior sagittal sinus. All right. So again, you can kind of see it up here at the top. There's the superior sagittal sinus. The inferior sagittal sinus would be somewhere in there. Okay. So the other type of dural septa is going to be found, all right, underneath the cerebrum, and it divides or separates the cerebrum from the cerebellum, okay? And so literally we call it the tentorium cerebelli, and I tell folks, think of a tent, all right? So this tent is going to cover, all right, the cerebellum, the mini brain, and it's going to separate the cerebellum from the cerebrum. And again, the tentorium cerebelli has a couple sinuses that are involved. It has the transverse sinuses, the straight sinus. Right, the transverse sinuses are going to be found on the side there. And then the straight sinus, you're going to find that in the center. I'll show you. I'm going to come right back. All right, so here are your transverse sinuses right here on the sides. So you can see, here's the cerebrum, here's the cerebellum. Okay, here's the cerebellum here in the sagittal cut, and then, well, we don't see the cerebrum up here because we took it out. But the tentorium cerebelli is right here. It's going to separate, okay, the cerebrum, specifically the temporal lobes and the occipital lobe, from the cerebellum down here. All right, and then you can see the transverse sinus here, and then it's tough to see, but here's your straight sinus. So basically, the inferior sagittal sinus goes from front to back as it's going back. All right, the inferior sagittal sinus then drains into the straight sinus back through here. We'll label these, not to worry. All right. Another dural septa is the falx cerebelli. This divides the two hemispheres of the cerebrum, or excuse me, of the cerebellum. Okay, so kind of like the falx cerebelli, or excuse me, the falx cerebri. All right, the falx cerebelli is going to divide the cerebellar hemispheres, and this is where we're going to see the occipital sinus. All right, I'll show you right here. Here's the occipital sinus here. And it's really hard to see the falx cerebelli, but it's right over here, small and tiny. It's going to divide, all right, the right cerebellar hemisphere from the left cerebellar hemisphere. And the last dural septa is the diaphragma cellae. All right, remember the sella turcica, that little saddle-like structure that sat, all right, in the sphenoid bone, and I told you that the pituitary gland would sit down in there. Okay, and it's got this little stalk here. It's called the infundibulum. Well, anyways, you've got this little bit of Dura Mater that sits right across there, and we call that the diaphragma cellae. It creates like a roof, all right, or covering over the pituitary gland. There's a little hole in it, so the pituitary infundibulum can poke through and get up there towards the hypothalamus. Okay? Like I said, it's the little roof there. Okay, so the next anatomical structure I want to talk about are the ventricles. The ventricles are these spaces or cavities within the brain. Now, this is important that you know this because it's in the ventricles that we make the cerebral spinal fluid. There's a special cell there, and we talked about those briefly, called ependymal cells. Essentially, what the ependymal cells do is that they kind of filter out the blood plasma, all right, and they help to make the cerebral spinal fluid. So it's these cells that are responsible for the creation of the cerebral spinal fluid. All right, so there's going to be 4 of them that you have to know. Okay, you've got the right and left lateral ventricle. All right, and then you've got the 3rd ventricle and the 4th ventricle. And we'll go through all those, okay? But again, these are just spaces or cavities in the brain. And they're filled, all right, with the cerebral spinal fluid. And we'll talk about what the cerebral spinal fluid does here in a couple slides. All right, so here you go, 4 ventricles, okay? You have 2 side by side, one on the right, one on the left. So we call those the lateral ventricles. All right, and they're separated by this structure here, which you do have to know for lab identification purposes. It's called the septum pellucidum. All right, they separate the left lateral ventricle from the right lateral ventricle. Then both of those ventricles are going to drain down into the 3rd ventricle. All right, that's located in the diencephalon. That's going to be central to your brain there. Okay, and so the left lateral ventricle has its own little tube that goes down into the 3rd ventricle, and then the right lateral ventricle has its own tube. All right, and so we'll see there's a connection there, all right, called the interventricular foramen, or also known as the foramen of Monroe. Okay, and then the 3rd ventricle will then drain into the 4th ventricle, which is right behind the pons. Okay, which is down in the midbrain. Okay, the pons is in the midbrain. The pons is directly in front of the cerebellum here. Okay, and it looks like, if you're looking at it from behind, it kind of looks like a diamond. The 4th ventricle does. Okay, and so the 3rd ventricle is connected to the 4th ventricle by the cerebral aqueduct. It's this long tube-like structure. So the cerebral spinal fluid flows down into here. Now, if it keeps going down, it's eventually going to wind up into the spinal cord. But also, it has another choice. It can flow out to the sides. And if it goes out to the sides, all right, if it goes both medial and laterally, all right, then it will circulate around the actual circumference of the brain there. And we'll talk about that later on. But the bottom portion, if we keep going inferiorly, that's going to take us down to the spinal cord. The spinal cord has us in the center of the spinal cord. It's tough to see, but if you look, there's a little hole there, right there. It's called the central canal, and that's where your cerebral spinal fluid, all right, it'll drain down into that, but also the meninges, remember, all right, in the subarachnoid space of the meninges, you also have cerebral spinal fluid. So you have cerebral spinal fluid at the center of the spinal cord, and then you have it between the pia mater and the arachnoid mater in the subarachnoid space that also surrounds the spinal cord. Okay? Please don't forget that. All right, so here's kind of what our ventricular system looks like. Okay, so you have these spaces. Here's our lateral view. So you have the 2 lateral ventricles here. All right, they drain down into the 3rd ventricle right here. And the third ventricle is surrounded by the diencephalon. Now, we haven't talked about that, but that's a region of the brain there. Then the third ventricle drains down into the fourth ventricle. And the fourth ventricle can drain down into the spinal cord, or, all right, the cerebral spinal fluid can go out to the sides here. Then it'll go circulate around the brain. Okay. So what is the cerebral spinal fluid? All right, CSF. All right. In a normal sample, it should be clear and colorless. Now, if you take a sample of cerebral spinal fluid and it's cloudy, you should be thinking bacterial infection. Analyze the bacteria. God forbid, if it's a bacterial meningitis, that's the worst of the two types of meningitis. There's viral meningitis, there's bacterial meningitis. If you have to have one, you want the viral. Okay? All right, so cerebral spinal fluid, like I just said, it circulates within the ventricles and the subarachnoid space. So it'll circulate throughout the internal portions of the brain and spinal cord, and then it'll circulate on the outside of the brain and spinal cord. All right, a couple functions. This is one of my favorite stats in this class, is that it provides buoyancy to the brain and it reduces the brain's weight by 95%. I would love for that to do that to me. My overall weight, and I could jump really high, but I don't have that luxury, right? So that's why I joke around and say if we didn't have cerebral spinal fluid, that your brain would fall right out of the foramen magnum, all right? It wouldn't, it would just clog it up though, all right? There's an actual condition called Arnold-Chiari malformation. Has anyone ever heard of that? Where your brain starts to sink into the foramen magnum, and then you start to have some neurological issues going on. All right, protection, yes. It's in a liquid environment. It sloshes around in a liquid environment. So if you jostle your head around, all right, that liquid sloshes around. I mean, it's anchored in there too for other reasons, but again, it's another layer of protection, all right, from damaging your brain, okay? Acts as a cushion, all right? And then amongst many of its other functions of environmental stability, what's it do? Well, it's going to transport nutrients and wastes to and from the brain. All right, and it helps to govern, for a perfect example, you've got these specialized receptors that are bathed in the cerebral spinal fluid, and they're called central chemoreceptors, and they monitor how much carbon dioxide is in your cerebral spinal fluid. If you have increased amounts of carbon dioxide, then most likely your pH levels will start to decrease, meaning, all right, your pH values will start to become more acidic. And when you start to drop into acidic levels, then you can get a very bad problem called denaturation of proteins. Your proteins can start to unravel and break down. And when proteins start to break down, you know what proteins do. They're involved, they're enzymes, all right? They act as voltage-gated calcium channels, those are proteins. Chemically gated calcium channels, those are proteins, all right? So proteins play a huge role in a lot of things. A lot of synthesis of enzymes, of secretions, of hormones, all right? So regardless, all right, when we talk about the environmental stability, it helps protect, all right, against those fluctuations in certain chemical environments there. All right, so how do we make cerebral spinal fluid? Okay, we have to have this structure here, the choroid plexus, and those are found in the ventricles, okay? And I'll show you a quick picture here. I'll come right back to the slide. So here you can see, all right, here's the choroid plexus. It's just a grouping here of these ependable cells. And if we zoom in even further, you can see here, you've got a capillary here, and then it's lined by these ependable cells. And so these ependable cells will literally just kind of filter out the blood plasma. All right, your red blood cells will stay in here, but it'll filter out the blood plasma, all right, outside of the blood vessel, all right, and that blood plasma will then be used for our cerebral spinal fluid. Okay, so again, we will, during that process of filtration, it helps to modify, all right, and we'll see with the cerebral spinal fluid, check it out, more sodium, that's awesome, all right, and chloride. Okay, because we already understand how the action potential is governed. Remember, all right, sodium and chloride, all right, salt water, banana floating in the ocean, all right, you're going to see quite a bit of that in the cerebral spinal fluid. Helps provide, all right, the ions that we need for action potentials, okay? So we'll have way more, all right, sodium and chloride in it, but you'll see we'll have a decrease in the amount of potassium. Okay, that's fine. You want less because you want the potassium to be inside the cells, all right? We'll see decreased levels of calcium, no problem, and then glucose, okay? All right, we'll talk about how we circulate here the cerebral spinal fluid. All right, but what will happen is, I think I got, oh, I do, okay, cool. Cerebral spinal fluid, all right, is constantly circulating throughout your central nervous system, all right? And so the levels of it or the amount of it is very sensitive. Has anyone here ever had cerebral spinal fluid drawn from them? Yeah? Did you ever get a headache afterwards? Oh, okay. Well, a friend of mine, when she had her baby, they gave her a lumbar puncture, but obviously not a lumbar puncture, excuse me, they had to give her an epidural. And in order, when they did that, they penetrated into the subarachnoid space, and so she was leaking cerebral spinal fluid out. And cerebral spinal fluid is the volume of it is so sensitive that a small change, an alteration, will cause massive severe headaches. That's just how sensitive that your body is to it, which was fascinating to me because I didn't know they did this, but they actually used her own blood and made like a blood scab. They would use their own blood and they put it on the puncture wound there of the arachnoid mater, and with the platelets and all that, and it would heal up, but it just wasn't holding. I think she had to have it done like 3 or 4 times, but she was getting wicked, massive headaches. But most of the times when people go in for a lumbar puncture, that's one of the side effects that they'll have is afterwards. Well, we're constantly circulating the cerebral spinal fluid through your central nervous system there, the meninges. And so there's going to be cases in which your brain makes too much. The ependymal cells make too much. So we have to get rid of that excess cerebral spinal fluid, and we drain it off into the dural venous sinuses. All right? So we'll see, we have these special structures called arachnoid villi, and they look almost like a mushroom cap. Okay? I'll show you a picture. My drawings are crappy. All right? And so the cerebral spinal fluid would come up into the mushroom cap. There's 2 cells in there. It pushes the cells apart so it can leave, and then when the pressure decreases, those 2 cells flop back down. So they kind of shut off the valve there. All right? So we'll see a big collection of these. We call those the arachnoid granulation here. I'll show you. Oh, no, okay. Now, let me show you this first, and then I'll backtrack here. Okay? So this is kind of what I was telling you about. All right, so here's the subarachnoid space. Okay? So the cerebral spinal fluid circulates through here. Now, when the pressure builds up, okay, the cerebral spinal fluid will move up here through the arachnoid villi, and then it pushes out into the dural venous sinus space there. Okay? And then when the pressure falls, these cells will close down. So the cerebral spinal fluid can only leave. It can't come back in. That's okay, because guess what? Cerebral spinal fluid is very similar to blood plasma. Well, this deoxygenated blood, this venous blood, is going to go back into circulation. And the kidneys will filter it, and then it'll just get recycled. I'm telling you, your body, when you get into 211, it's going to blow your mind. Your body is awesome at recycling things. It's like the ultimate recycler. If you think you're a good recycler, you ain't got nothing on your body, because it uses stuff all the time. Okay? So that's what we're going to see. We're going to see it get pushed out through here and into, the dural venous sinus space, and then just go back into generalized circulation and circulate through the body here. All right. So what we're seeing on this slide, okay. All right. So you can kind of see here in our ventricle. Here's where the 3rd ventricle is. All right. The 3rd ventricle drains down here into the 4th ventricle. So that cerebral spinal fluid can go down here and down into the spinal cord if it needs be, or it can go out laterally, and then it'll circulate around the brain. Well, if it goes out laterally immediately, it's going to go here into that subarachnoid space here and circulate throughout. And then eventually, as it circulates throughout, if it finds those arachnoid villi, it just pushes those arachnoid villi open. All right, if the pressure of the cerebral spinal fluid increases, and it drains the excess fluid out into, the dural venous sinuses there. All right, I'm going to stop there, and let's do some labeling, and then we'll take off. It's a good place to stop because I love the blood-brain barrier. It's really, really important to understand the blood-brain barrier. We can really learn it later. All right, so if you want to bust out your models or your atlas, we're going to go through, okay, the anatomical structures of the brain. Okay, so let's start off with the first region of the brain that we talked about tonight, okay, the cerebrum. So if you look here, again, the cerebrum is this whole structure that has all those folds, all those little indentations, all right? That is going to be your cerebrum, all right? This is the lateral view here. Remember, there's 4 regions to your brain. Cerebrum, diencephalon, all right? Brainstem, and the cerebellum. Now look at this slide here. Watch the arrow. I'm going to change the slide and that arrow will never move. Boo. Well, it kind of did. Wait, that's the wrong. Oh, no, that's the right one. All right. Actually, it's just a different slide. All right, hold on. Let me go back up here. Let's not go out of order. All right. So now we're looking at the posterior view. This is the posterior view because I can see the cerebellum. The cerebellum's on the back. Okay, so this is anatomically correct. So we're looking, it's like if I were standing behind you in line, this is what I'm looking at. So here's the left cerebral hemisphere. Okay. Then on the other side here, you got you guessed it, the right cerebral hemisphere. And they're separated by the longitudinal fissure, but I'll show you that in a second. Okay, so there's 4 visible lobes to the cerebrum. All right, first one is the frontal lobe. All right, we talked about all its functions: motor function, all right, higher order functions, personality, intellectual thinking, reasoning, decision making. Okay, so all that. So that's the frontal lobe. Then you got the temporal lobe. Hearing and smell is the temporal lobe. Okay. Then superior and posterior parietal lobe. So again, keep in mind where the cranial bones fall into place here, and this kind of follows that pattern there. All right, parietal lobe is going to be mainly made up of sensory activity. And then finally, where all our visual functions hang out, occipital lobe on the back. All right, occipital lobe. Do yourself a favor, make sure when you take your lab test to see if there's any question on there that asks you to answer the question or region, okay? If it's lobe, obviously, it'll set, you'll put that down. But if it's region, then you would, if that arrow is pointing to that area, you would say cerebrum, okay? Just be careful. This one you don't have in your atlas, that's the insula, okay? So we just kind of pulled the temporal lobe down, and you can see the insula right in here. All right, now we're going to talk about some of those cracks and structures and fissures and whatnot. Here's the longitudinal fissure. Okay, that's that space or that deep depression there in between, okay, your right and left cerebral hemispheres. Longitudinal fissure. Now, in the longitudinal fissure, we just talked about this about 10 minutes ago, right? You are going to have the falx cerebri. That's that dural septa, okay, that Dura Mater that's fused together that kind of is in that space to separate the right and the left. I'll show you a better picture of this arrows in the line are just representing the space in which the falx cerebri is occupying. Oh yeah, do I have it in here? Nope, I don't have it in the slideshow. The next slideshow we'll go over it though. All right, but it is an actual thing. Yep. All right, so the transverse fissure is that horizontal fissure, okay, that's found in between, all right, your cerebrum and your cerebellum. Okay, this line is occupying it. You can see the space better over on the right side here. Okay, that's the transverse fissure. So again, in the transverse fissure, you're going to see that Dura Mater, all right, that dural septa, excuse me, let me be more specific. And the name of the dura septa that sits in there is called the tentorium cerebelli. And again, it's like a tent flap. That's what I tell folks. It just lays right on top of the cerebellum.
Are those the same two areas?
Yeah, also it's just it's the same area. One is a space, and then the other one, like this slide, is just telling you what is occupying that space. Okay. Oh yeah, oh yeah. I'll show it to you. It's not on this. It's not in this slideshow. Well, next what's today? Thursday. So on Tuesday, we'll go over those because we're.
Or identification, is it just going to be the transverse fissure, or will we need that what's in there?
It can ask you both. Yes, you have to know the fissure, and you have to know what could be in there. There's better slides where you'll actually see the tissue. Okay, this is just asking you if you know what's in that space. Okay, so back to the lateral view here. Okay, you have this structure here. All right, the lateral sulcus. But this is what I was telling you in lecture. All right, it's also referred to as the lateral fissure. Okay, either or is acceptable. All right, and the lateral sulcus or lateral fissure is that space that or that structure that divides the temporal lobe, all right, from the frontal and the parietal lobe. So here's the frontal lobe over here, and then the parietal lobe's over here. Okay, so you see this kind of deep, it's not deep, I shouldn't say that, but it's very prominent. This very prominent, all right, sulcus. All right, that's a huge anatomical structure that is the central sulcus. Okay, that black line is in that space. The central sulcus, all right, is the anatomical structure, landmark, that divides the frontal lobe from the parietal lobe. Parietal lobe's back here, frontal lobe's here. Okay? So the gyrus, all right, that's in front of the central sulcus, that's what this next slide is pointing to, that's the precentral gyrus. That's your primary motor cortex. Okay? So as you're writing and labeling things down, all right, the first, all right, nerve signals are generated in that space, in that region there. Okay? That's in front of the central sulcus. Now, behind the central sulcus is this gyrus, and that's the post-central gyrus. And that is your somatosensory cortex. Okay, so sensory information, conscious sensory information, right? If you're cold, that information is reaching that area. Okay, if you're sitting on something hard, that information is reaching that area there. All right, we're going to move on to the next region of the brain. All right, for our labeling, there it is, cerebellum, the little brain. Okay, lateral view here, you can see it's all right, inferior to the cerebrum, and it's posterior, all right, on the brain. Sits on the back. All right, so this is a posterior view here. So this region right here in the middle, okay, is the vermis. And it's that connection between the two hemispheres here. It's the vermis. All right. So to the right of the vermis, this structure here, that's the right cerebellar hemisphere. And then obviously on the left side, there's the left cerebellar hemisphere. Okay, so now what we're going to do is we're going to do a mid-sagittal cut the brain straight down the middle. Okay, now remember what we said. In the brain, gray matter's on the outside, white matter's on the inside. So we give the white matter on the inside of our cerebellum a special name, arbor vitae, tree of life. You might not have this one, but you definitely have this one. All right, arbor vitae. That's that white matter on the inside. All right, let's move on to the diencephalon, the third region of the brain. Now there's 3 parts to it, okay? 3 parts. And we'll go through each one individually. I just want to show you, all right? All these arrows are pointing to 3 different structures. So the diencephalon is made up of the thalamus, the hypothalamus, and the epithalamus. And part of the epithalamus is the pineal gland. All right, so we're going to go through each of these individually first. All right, here's the first one. There's the thalamus, that pinkish kind of football-shaped structure, this whole thing in here. All right, that's the thalamus. Okay, here it is on this model here. Thalamus. Okay, so that's the first part of the diencephalon. The second part is the hypothalamus. That's this region here. Yep, it's funny because that's what I think too. Yep, and I know some of my colleagues call it like a duck's bill. Okay, that's the hypothalamus. And it makes sense. Hypo is below. Low or inferior to the thalamus. All right, you can see it here on this model. This one here definitely looks like a duck. Don't you feel like this is like a mallard? Like here's the head, here's the eye, here's the bill down here. Looks like a duck. And then you've got the neck and all that down and through here. Hypothalamus. Okay, then the last part is the epithalamus, but again, we're only going to have you label the pineal gland, which is this kind of little swelling out pocket back here, that little pinkish region right there. All right, pineal gland is on the posterior portion. Okay. And this is the gland that helps you sleep at night, produces melatonin. All right, here you can see the pineal gland on this one. All right, so if we're still thinking that this structure looks like a duck and the duck's bill here on the front, all right, you've got a structure there that has to do with vision, and that's called the optic chiasm. Basically, you have an optic nerve that travels from the eye. So you have one for the right eye, one for the left eye. And where they kind of go backwards from the eye and they join each other and there's a crossing over of nerve fibers. Where they join each other is the optic chiasm. The optic chiasm, if you look closely, sits above this thing here. All right? That's the pituitary gland. The pituitary gland is part of the endocrine system, okay? And that's the gland that sits in the sella turcica. So the pituitary gland is connected to the hypothalamus. You can't really see it on this model, but here's the hypothalamus. Here's the pituitary gland. It's connected by this little thing here, all right? It's known as the infundibulum. Right, that's the structure that connects the hypothalamus to the pituitary gland. Sometimes the pituitary gland can get a tumor growing on it. We call that a pituitary adenoma. And as that gland or that tumor gets bigger and bigger and bigger, it can press on the optic chiasm. And I actually had a patient last semester, a student last semester, whose mother had a pituitary adenoma, and some of her first symptoms of this pituitary adenoma, and this is something that we talk about when I was in med school, is that you start to lose your peripheral vision. And she was textbook pituitary adenoma because it pressed on the optic chiasm. Okay, so we've got our commissural fibers. This is the corpus callosum. This connects the right cerebral hemisphere to the left cerebral hemisphere. All right. So it's this whole structure right here, all of that. That's the corpus callosum. Okay, you can see it here on this model here, corpus callosum. All right, a couple other structures that we find in there. We got this blue colored structure, and that's the choroid plexus. And we just talked about that. The choroid plexus is that grouping, all right, of ependymal cells. All right, you'll have capillaries there, and that's what makes our cerebral spinal fluid. All right, the choroid plexus. Now, when we label the ventricles next class, all right, remember what I said. Between the left lateral ventricle and the right lateral ventricle, there is a partition. And that partition that separates both of them is called the septum pellucidum. Okay, it's this barrier in between the left and right lateral ventricles there. And then you can see it on this model here. Now, near the end of this chapter, chapter 13, we are going to talk about the limbic system. The limbic system is the emotional system of your brain. Okay, it involves mainly emotions, but there's some other functions to it. And one of the structures that are part of the limbic system is this guy right here, this little white kind of streak there. That's the fornix. It's superior to the choroid plexus there. Also superior to the thalamus. You can see it over here on this model. Okay, the last part of, the last region of the brain is the brainstem. There's three parts: the midbrain, the pons, and the medulla oblongata. So the first part here is the midbrain. Now it's kind of tough to see, all right, but basically the midbrain is pretty much from here to here, between those two lines, from here to here. All right, you can see, so there's a space that runs right through the middle of the midbrain here. That's called the cerebral aqueduct. Okay, but some of the midbrain is back here too, here, and here's the anterior portion of it. All right, so on this back portion here, okay, this white structure, we're seeing it now. If I were to zoom in, you can't, this model's not the best, but if you get close to it, you'll see this. It'll look like this from the lateral view. See these two bumps coming off, here and here, okay? Now, if I were to turn this thing and look at it from the back, so if I were to look at it from the back here, I'm looking at it from the side, but if I were to turn this structure here and look at it from the back, it would look like this. Looks square, and it'd have four bumps coming off the back, okay? Call that the tectal plate. All right, but now we're only going to be looking at it from the side view. So this next slide is pointing to those top two bumps. Okay, we're only going to see it, so they're pointing to those top two bumps. So we call that the superior colliculi of the corpora quadrumina. That's a mouthful. That's a tough one. That has to do with visual eye reflexes. But it's those two bumps. So if I were to be looking at that same structure from the back, all right, we have our four bumps on the back. It's the top two. Okay, those two top. Okay, so now what we're going to do is we're just going to drop down and point the arrow to the bottom two bumps. That's the inferior colliculi of the corpora quadrumana. That has to do with auditory reflexes. All right, y'all, almost done. Okay. All right, midbrain from a different model here. You can see, there you go. Just to keep an eye out, there's the midbrain here between those two blue lines. All right, so the next part of the brainstem is the pons, means bridge. Okay, it's this fat swelling here. It's directly in front of the cerebellum, right there. Okay, it's in the middle. And then the last part, which is one of the coolest parts of the brainstem, all right, is the medulla oblongata. Okay, it's the last part of the brainstem before we get to the spinal cord. Medulla oblongata. A lot of important regions for vital functions of your body. There it is on that model. Here it is on this model. That's the last slide. Booyah. Okay. That's it for the labeling. We will do the ventricles and we will do the dural septa on Monday. Wait, Tuesday. Sorry, Tuesday. Today is Thursday. Nah. Okay. That's it. That concludes